Hello, my name's Hannah Ashraf. I'm a junior at the Altamont School, and my project is the effect of checkpoint inhibition on modulating the hypoxic tumor microenvironment. Lung cancer is the leading cause of cancer-related deaths in the world, accounting for 1.8 million deaths in 2018 alone. There are two main types, small cell lung cancer and non-small cell lung cancer, or NSCLC. We worked with an adenocarcinoma NSCLC cell line, and NSCLC accounts for around 87% of all lung cancer diagnoses. Despite screening methods, most patients are diagnosed with metastatic disease, which is why the 5-year survival rate is so low at only 23%. Traditionally, cancer treatment relies on chemotherapy and radiation, which kill rapidly dividing cells in the body. However, patients often become resistant to chemotherapy. Immunotherapy is a relatively new treatment which helps the body's own immune system to recognize and attack cancer cells. One form of cancer immunotherapy, called immune checkpoint blockade, involves blocking the interaction between a protein found on T cells called programmed death 1 or PD-1 and its ligand, programmed death ligand 1 or PDL1, which is found on many tumor cells as well as tumor-promoting cells. The tumor hijacks the immune checkpoint mechanism of the PDL1 pathway, which is used by normal cells to evade attack from immune cells. When PD-1 on the T cell binds to PDL1 on the tumor cell, the interaction between the two blocks the T cell from being able to attack the growing tumor, as well as causing apoptosis of the T cell. We can block this interaction, however, by using an anti-PDL1 blocking antibody. However, PD-1 is not the only pathway that causes resistance to treatment of cancer. The tumor microenvironment, or the TME, is composed of many different biological components. We focused on tumor cells, stromal fibroblasts, and T cells. A key contributor to treatment resistance is hypoxia, which is a low oxygen level. Hypoxia makes cells grow slower, which causes resistance to chemotherapy and radiation because they attack rapidly dividing cells. Hypoxia can also directly increase the expression of PDL1 in tumor cells by directly binding to the hypoxic response element, or the HRE, in the promoter of the PDL1 gene. The immune checkpoint blocking antibody can interfere with this PD-1-PDL1 interaction, but the impact of this inhibition on the hypoxic lung and tumor microenvironment is unknown. So we hypothesized that by targeting the immune checkpoint PD-L1-PD-1 interaction via a blocking anti-PD-L1 antibody will impact the biochemical microenvironment within the tumor by both decreasing hypoxia as well as inhibiting tumor growth. So to test our hypothesis, we have developed a 3D tissue model, which is called a bioreactor. And so it's made out of a clear silicone polymer called polydimethylsiloxane, or PDMS, and it contains a central chamber in which cells are co-cultured within an extracellular matrix, or an ECM, containing 90% collagen type 1 and mixed with 10% low growth factor matrigel. This central chamber has steel wires, and it generates these through channels that are created following the ECM polymerization. So when the wires are removed, cell culture media can pass and flow through the channels to deliver nutrients. Following setup, the bioreactors are moved to a 37 degrees Celsius incubator, where they are connected to a peristaltic pump for 10 days. The media is changed every 3 days, and circulating cells and media are collected and stored. The perfusion rate is the rate at which the media passes through the channels, and we set it up to try and mimic the microvasculature in the body. The size of the bioreactor as well is meant to mimic the real size of a tumor in the body. So this 3D model was used to co-culture 3 different cell types. We had our lung cancer cell line H358, which we labeled red using the lipophilic dye PKH26. We also had the lung fibroblast cell line IMR90, which we labeled green using the lipophilic dye PKH67, and we finally had our unlabeled JERCAT T cells. To test my hypothesis, I wanted to compare a treatment with either an IgG control or an anti-PD-L1 antibody, both at 10 micrograms per milliliter, and 7 replicates for each experimental group. We anticipated that the anti-PD-L1 antibody will interfere with the PD-1-PD-L1 interaction. while the IgG control is a non-specific antibody that is used to show that our results are specific. We monitored hypoxia using a hypoxia probe called HypoxySense 680. It was injected into the bioreactor 1 day before imaging, and the hypoxia dye recognizes carbonic anhydrase 9, which is a protein that is commonly upregulated in hypoxic regions. So we had 3 main objectives. We of course wanted to evaluate hypoxia levels from our IgG control to our anti-PD-L1, as well as evaluating cell type specific changes in hypoxia, for example, specifically around the fibroblasts or the tumor cells. Our final objective was to monitor cell growth and cell density, and all of this was of course in the presence of an anti-PD-L1 blocking antibody. So for our first objective of evaluating changes in hypoxia in the tumor microenvironment, we wanted to measure global hypoxia levels, and to do this we used the IVUS Lumina. And so, as you can see in this first picture, it's a representative image of sort of the amount of hypoxia signal for a control group versus the anti-PD-L1. So as you can see, there's a lot more hypoxia from day 5 to day 10 for the control group versus from day 5 to day 10 of the anti-PD-L1. So we quantitated all of this in 7 bioreactors, and we saw a trend towards a reduced hypoxia in the treated group as compared to our control. We then used confocal microscope and Fiji software to image hypoxia at a cellular level, evaluating random regions of interest, or ROIs, within the engineered tumor microenvironment. So these are some representative images of the difference in hypoxia at a cellular level. The red represents cancer cells, the green represents fibroblasts, and the blue is a dye called Hoist, which stained the nuclei of all of the cells and was added at day 10 to be able to measure the signal for T cells as well. The white signal of hypoxia is much stronger at day 10 in the IgG control as compared to the anti-PD-L1 treated group. So the graphs show quantitation of regional hypoxia intensity measurements within ROIs in several different cells. We see a significant decrease in regional hypoxia at both day 5 with a p-value of less than 0.0001, as well as day 10 with a p-value of less than 0.01 with anti-PD-L1 treatment when it is compared to the IgG control, and there were 7 to 8 bioreactors for each condition. So then we wanted to quantitate the effect of checkpoint inhibition on hypoxia for all 3 cell types separately. So the first picture is a representative image of what this may look like. So as you can see from the arrows pointing towards the white signal, there is signal around the tumor cell, but not really around the fibroblasts. So we can draw these areas of interest around the cell. You kind of go like this. I'm not doing it very well. And so you can measure hypoxia signal for specifically inside of that region. And so for our results at day 5, we see that hypoxia levels decrease significantly in the anti-PD-L1 treated group as compared to the IgG control group. And this was for both tumors and fibroblast cells. So we see a pretty statistically significant decrease for hypoxia levels for day 5. So we also wanted to measure cell type specific hypoxia changes at day 10. And so for our day 10 results, we could also see the effect of checkpoint inhibition on T cells because we added that Hoist dye. So we saw that along with the tumor cells and fibroblasts, hypoxia in T cells was significantly reduced at day 10 following an anti-PD-L1 treatment as compared to the IgG control. This makes a lot of sense since we know that the PD-1-PD-L1 interaction causes T cell exhaustion as well as apoptosis. So the reduction in hypoxia levels in tumor cells and fibroblasts was also just consistent with what we saw at the day 5s. So our final objective was to evaluate the effects of anti-PD-L1 therapy on cell growth and cell density. And so we measure cell density as a way, like the number of nucleated cells per area, as a way to measure cell growth. So to evaluate this, following day 10 imaging, our 3D cultures were formalin fixed and histologic cross-sections were imaged using a digital microscope. A one-way ANOVA testing showed that there is a trend towards reduced cell density with checkpoint inhibition for this as well. So in conclusion, in our 3D lung tumor model, immune checkpoint inhibition by an anti-PD-L1 antibody lowers hypoxia of T cells, tumor cells, and fibroblasts significantly compared to our IgG controls. This treatment also shows a trend towards reducing cell density. Thus, modulation of immune checkpoint proteins may impact immunosuppression in the lung tumor microenvironment. Our 3D lung tumor models may be utilized to evaluate responses to immunotherapy strategies. Some possible future directions could be to evaluate the effect of the inhibition of hypoxia by an anti-PD-L1 antibody on T cell function. So to do this, we could label T cells with a different fluorescent marker and use flow cytometry to determine if reducing hypoxia changes T cell exhaustion by assessing change in PD-1 expression and other markers of exhaustion. Another future experiment would be to measure cell death in response to checkpoint inhibition by evaluating circulating LDH levels as well as monitoring apoptosis. Additionally, we could target PD-1, which also targets the PD-L1-PD-1 interaction, to confirm changes in hypoxia as well as cell density. Finally, I'd really like to thank my mentors, who I couldn't have done any of this without, Dr. Kelly Goliwas, Dr. Jessie DeShane, and Dr. Mary Williams. Thank you for listening.